Berkey is here interviewing for a faculty position in mechanical engineering. He's a postdoctoral fellow at the University of Colorado Anschutz Medical Campus. He received his PhD in bioengineering and mechanical engineering at Georgia Tech in 2010, where his research focused on computational biofluids in medical device development. He completed research fellowships in Australia and at CSU. Some of you, a lot of you probably know him already. So Dr. Burke's current research investigates how cells in microorganisms produce and respond to external forces in a flow environment with the overarching aim of developing diagnostics across species and therapeutics to benefit human health. Welcome.
Thank you very much. And thank you for inviting me. It's an honor to be here in front of all of you today. I know many faces, so it's nice to talk about my research. So today I'm mostly going to be talking about work I did here with Prasad Dasi, specifically work on developing prosthetic heart valves and also looking at development of the heart during their embryonic stage. So my big interest is really in biofluids, so it's important then to define what biofluids consist of. So this can be fluids in our lungs, so as we breathe, this is important, and then also the circulation of our blood is important to transport nutrients and expel waste. Lymphatic vessels are important for transporting immune cells throughout our body. And there's many other fluids that are important internally for our bodies. And also biofluids can really include locomotion of organisms through water or air. But my big focus so far has been on the cardiovascular biofluids, so blood flow. So you may be asking yourself why cardiovascular biofluids, why not a different fluid? And the main reason is because cardiovascular disease has remained the leading cause of death in the United States for over a century now. It's a big growing problem in developing countries as well, as we're figuring out how to cure some of the diseases over there, and also because they're developing Western diets. By the age of 80, 85% of us will have some form of cardiovascular disease. So raise your hand if you know someone that's had a heart attack, a stroke, heart arrhythmia. So these are all forms of cardiovascular disease. And even at the young age of 40 to 60, about half of us will have some form of cardiovascular disease. I know it's a big problem in my family. So that's really why I'm focused on this disease. But in addition to being a killer, it's also the leading expenditure for our healthcare system, even exceeding cancer. And this number isn't decreasing, it's actually expected to increase by these figures by threefold by 2020. And these are figures from 2011 and 2012. So hopefully I kind of emphasized the relevance and importance of cardiovascular disease. But then this brings into question, what is the cardiovascular system? So as mechanical engineers, we're not always working in this area, so I'll briefly kind of explain what the cardiovascular system is and how it works. So its main purpose is to supply nutrients such as oxygen to our cells, which our cells require in order for us to be alive. And it also removes waste efficiently from a number of our organs, and it also maintains a chemical homeostasis. And just briefly, the way the circulation works is you get blood returning from your body, and it enters the right side of the heart. At this stage, it's deoxygenated, and it has substantial CO2 levels in it. And then from here, it enters our right ventricle, which then pumps blood up into our lungs, where it expels CO2 and picks up oxygen. As I mentioned, our cells require this. And then it comes into the left side of the heart. And this is really the powerhouse of the heart, because it then has to supply blood to the rest of our body, which, as you can imagine, as mechanical engineers, there's quite a bit of resistance with our vasculature throughout our body. So it's quite a powerful pump. And then this blood can then be filtered through the kidneys and the liver. So you may then be asking, why is a mechanical engineer really interested in the cardiovascular system, and what can a mechanical engineer do in this area? And really, the cardiovascular system is a series of pipes and pumps, but it's also not as straightforward as a rigid circular pipe. So the system is always responding to its changing environment. If it didn't do this, it would be difficult for ourselves to stay alive. So amazingly, this system has an ability to adapt to changes in load. So one way is it senses the wall shear stress. So this is the stress that moves along the inner lining of our vasculature. And amazingly, our cells can sense this change in shear stress and then signal for changes in the vasculature in response to these changes. Otherwise, you can imagine if you're increasing flow to a particular part of the body, shear stress will start increasing, and that can start creating large stress on the cells, which can then create damage. But instead, the cells signal for dilation, and then you can reduce the shear stress, so it's not a problem. In addition, hoop stress is sensed by your cardiovascular cells as well. So we can think of Laplace's law or a cylindrical pressure vessel, and you can think of the vasculature as this. And it actually, the arteries maintain a hoop stress of about 10 to the 5th Pascal. So if you have hypertension, meaning that your blood pressure is high, your body actually responds by remodeling your blood vessels in order to accommodate for this higher pressure. If it didn't, you'd be at much higher risk of your blood vessels bursting. So our body is always responding to changes in load. And probably the most visual way we can see an example of changes in our body to loads is as people work out, their muscles get bigger, they're accommodating for this larger stress that our muscles are experiencing. And conversely, if you're an astronaut going into space, your musculoskeletal system doesn't see these loads, and so it can begin to deteriorate. So this is also a big concern for people going into space, or if you're going long-term into Mars as a fictional character. So a classic example of unfortunate response of our body to changes in load is the development of coronary or carotid artery disease, in which we get plaque growth or atherosclerosis. So it's been shown that there's a very strong correlation between low wall shear stress and atherosclerosis, and this was shown in the 1980s by my PhD supervisor, David Koo, and this work is still a popular topic at biofluids conferences, and this work has had over 2,000 citations. So in this case, plaque is growing and then can result in a heart attack or a stroke, and so it could be bad as well if you can adapt to some of these loads. And so this is an adaptation and a maladaptation to low wall shear stress. So as engineers, we want to know, where can this sort of thing occur? Well, if we really simplify Navier-Stokes down to just the inertial terms, we see that we get adverse pressure gradients where we have decelerating flows, so this first term with respect to time, or if there's a flow expansion. So this is why we see a lot of plaque formation in the carotid sinus, and why we see plaque formation in the coronary arteries where we get large oscillatory flow. So again, our body is quite responsive to changes in loads. And so with that, how can we investigate the cardiovascular system? As engineers, we like to use analytical or computational models to understand the physics of the system. We also can use in vitro experiments. So for example, you can remove the bone from the body and begin to perform tensile test or compressive test on the bone to then evaluate the properties of it and how it changes with disease. And with these, you have a lot of control over the system, which we like as engineers. In biology, there's a little more focus on in vivo experiments, which means that they're doing experiments of within the body. And this is the natural state that our cells experience and parts of our body experience. So this can also be equally useful, and is also the focus of clinical research. So if you have a clinical trial, usually they're testing out a new drug or device, on a patient, and so their big interest is on the in vivo experiments. But it's really with these combined tools that we can really understand the system and start treating the disease. And so I've used most of these tools in my research. One topic that I'll talk about today is using these tools and looking at embryonic heart development and how the heart responds to changes in load as it develops. I've used 0D and 1D computational models to look at how potentially flow and pressure waveforms flow going through our cerebrovascular system can indicate whether someone's got a risk for Alzheimer's disease. In addition, used in vitro experiments to understand how fluid can influence the stresses on our heart valves, which I'll describe a little more. And then in addition to stress on our cell lining of our cardiovascular system, there's stress that can also be experienced by red blood cells and cells that are involved in the flow component. And so turbulence is a big concern in that regard, and turbulence can cause cell damage or hemolysis, meaning your red blood cells are rupturing due to stress, and it can also induce platelet activation, which creates a blood clotting response. And speaking of the blood clotting response, our research is also focused substantially on thrombosis, which is a blood clot that generally kills us when we have a heart attack or stroke. So plaque forms over decades, whereas in the instant that you get a rupture in your plaque, your blood clots and forms this thrombotic response and then blocks flow to your vital organs like your heart or your brain. And then I also have a strong interest on the translational side of developing cardiovascular medical devices to treat some of these diseases. So if you have plaque growth or atherosclerosis, you can expand a stent to then create normal flow again through that particular vessel. And so the problem with these stents is they create regions of low wall shear stress and also regions of high stress that can either result in a poor endothelial response for the low wall shear stress, or it can result in thrombosis. So I've looked into the fluid mechanics around stents. In addition, I've looked at developing prosthetic vein valves. So as I stand here, you can imagine there's a giant column of fluid that's creating hydrostatic pressure in my legs, and these vein valves really help distribute that pressure so that it doesn't build up as I'm standing here. When these valves fail, pressure builds up and you can get varicose veins in the case of superficial veins on your leg, or in the case of deep veins, valve failure can actually result in deep vein thrombosis, which can then embolize and create a thrombus that goes into your pulmonary system, which can be quite problematic. So looked at developing prosthetic vein valves as well. And then here at CSU, I worked a lot on the development of prosthetic heart valves, which I'll talk a little bit about today, and this was work I did with Prasad here. So the heart is at the center of the cardiovascular system. It's going to be the main topic for today, and this has been of interest even back to the days of Leonardo da Vinci, where he's trying to understand the flow patterns around heart valves and around the heart. So first, when talking about the heart, I'll explain how it develops and how disease can result from development. So the heart's the first functioning organ. It begins as just a simple tube, and at this stage it has no valves, it doesn't have any clear chambers, and yet despite this it can create a fairly unidirectional flow, pumping blood throughout the embryonic body. After this tube stage, it begins to develop some chambers that begin to form, and pumping slightly changes, and then eventually it begins to loop, forming more distinct chambers that pump independently, and eventually you end up with a multi-chambered adult heart that has fully functioning heart valves. And then to study this, there's various animal models that have been used, including mice, frogs, chickens, and zebrafish. And what's been found is actually if you impair flow or modify flow in the heart while it's developing, you actually get poor formation and development of the heart. And this can be problematic because it could result in congenital heart defects. And congenital heart defects are actually the leading cause of cardiac arrest in people under the age of 13. So you can imagine this is quite a problem. It's always sad when the child has a disease. So to study this, we really want to understand how the body is responding to the mechanical environment during development. This is because only 20% of the congenital heart defects can be attributed just purely through genetics. So that means somehow 80% of these other defects must be coming from something. And we believe it's due to the environment, and specifically we think it could be due to the mechanical environment. And so initially, as the heart's developing, you have genetics really controlling the cardiac structure. So the myocardium, which is the muscular layer around the heart, the endocardium, which is the inner lining of the heart, and also the cardiac jelly, which is a gelatinous layer between those two layers. And with the structure, it creates pumping and pumping mechanics. And depending on how the pumping mechanics occur, you get blood supply to the body, and this results in wall shear stress and circumferential stress or hoop stress. And then, as you imagine, if we don't have feedback of that stress to this developing system, then the body can't respond to any perturbations. So as mechanical engineers, again, we want to understand this feedback loop and understand what role it has in these congenital heart defects. So to study this, we're using the zebrafish, and this is work that I'm doing with Deborah Garrity. And zebrafish have a number of advantages over some of the other models, and probably the most advantageous advantage in the case of zebrafish, in my mind, is the fact that it can obtain nutrients and specifically oxygen through diffusion without flow of our cardiovascular system. So it allows us to more isolate mechanical effects of changes in flow relative to the impact of nutrients if we do any flow manipulations. So with that, we can perturb this system by modifying genetics, or we can apply drugs, surgery, or flow manipulation to then impact the feedback loop to better understand how alterations could impact congenital heart defects. So to understand that system, we really need to quantify flow and pressure so that we can quantify stress in the body as it's developing. So one way to quantify flow is through particle imaging velocimetry, which is a typical experimental fluid mechanics approach to quantify flow. And we use red blood cells as the seeding particle in this case because it's not invasive. There's some other techniques that our group has developed as well to look at this. And then once you have flow, you can actually go to your momentum equation and Navier-Stokes, and you can then estimate what that pressure distribution is as well inside the heart as it's developing. And that's just based off of your gradients and velocity. So if you have your velocity gradients through PIV or particle imaging velocimetry, you can estimate the pressure distribution and therefore circumferential stress. So in addition to quantifying flow, we want to perturb the system somehow. So one way we've done this is systemic perturbation by putting the zebrafish through this amusement park ride where we centrifuge it and all the blood pools to the zebrafish's head. And we allow this to happen for a 6-hour period. And with that, you don't get normal flow through the heart, but then you can evaluate how the zebrafish then responds after it's been through this system. So on the left side, you can see a normal heart through its looping stages where you get some independent contractions of the atrium, which is at the bottom, and then also the ventricle that's at the top. However, for zebrafish that spun, just as the heart's developing, you don't get the same volume of blood flow coming out through the heart. But this is a specific problem that seems to only work when you spin the fish at 24 to 36 hours after it's been fertilized. And this imaging occurred at 48 hours, so there's been time for the zebrafish to recover. If you spin it at other time points during development, it doesn't have the same effect on blood flow. So we can use our quantification techniques to then quantify blood velocity and pressure. And with that, we can estimate what wall shear stress is and what circumferential stress is, which are the stresses that can actually be felt by the cells, since they can't sense what's going on inside the blood itself. We find that the spun fish ends up producing a lower pressure gradient and therefore a lower flow as it brings in blood through the body through a suction mechanism as the atrium expands. And then also there isn't as high of a pressure gradient on the spun fish as the atrium contracts, sending blood into the ventricle. So again, blood flow is lowered and therefore you don't get as much blood volume getting pumped through the body. And so this fish has been adapted from the spinning. And what we found is that what's really controlling this pressure gradient is related to the rate of contraction of the atrium and of the ventricle, and the rate of expansion as well. And then also viscous resistance between the two chambers. And the rate of contraction we're finding is actually lowered in the spun fish. And we think it's lowered because of the poor development of this jelly-like layer that exists between the other two layers of the heart. And so it can't do normal proper pumping. And we believe that maybe the cardiac jelly is not forming in response to this altered flow environment during early stages of development where it begins to form. So in addition to early development, we're also interested in adult heart valve disease and how our adult hearts might misfunction. So this is the heart with all four chambers, right side of the heart with an atrium and ventricle, and the left side of the heart with an atrium and ventricle. But it's really the left side of the heart that ends up with most of the disease. And this is, as I mentioned earlier, where a lot of the contractile power is coming from, is the heart sends blood throughout the body. So in terms of the valve that enters from the ventricle into the aorta, which feeds blood to the rest of our body, you can end up with an aortic stenosis, which is results in an improper opening of the aortic valve. So this requires a lot more pumping effort of the heart compared to the normal operating heart valve, and also can cause a risk for thrombosis and blood clot formation. In addition, the mitral valve suffers from potential reverse flow, so the valve no longer allows flow to go in one direction. Now there's flow known as regurgitation that can come back up through the valve, and this can be caused through a number of problems within the heart. So one approach we have to treat these failing valves is to apply a prosthetic heart valve. And this has been a big focus of my work over the past couple of years. And over 280,000 heart valves are implanted worldwide each year. They can come in the form of a bioprosthetic valve, which means that these valves are formed of a biological material. The disadvantage with these bioprosthetic valves is they suffer from durability issues, usually associated with something known as calcification, so they essentially become more rigid compared to their initial tissue. So alternatively, there's mechanical heart valves, and so as engineers we can engineer durability into these valves. Unfortunately, they result in hemolysis, which is the rupture of red blood cells, and also thromboembolic events, so formation of these blood clots on the valves that can then dislodge and create blood clots in our cerebrovascular, creating a stroke, or can result in clots moving into other vital arteries. And so one way clinicians prevent that from happening is they'll apply a drug regimen where they'll give anticoagulants to the patient so they don't get this thrombus formation or blood clot formation. The problem with applying these drugs is the clinicians are really trying to do this fine balance between someone being at risk for thrombosis, which is highly deadly, or at risk for bleeding, which can actually also be quite deadly, especially if you have to go into surgery. So there's this fine balance that clinicians are constantly trying to make when these patients receive these valves. So alternatively, we can look at designing these valves differently so they're less prone to thrombosis. And one way we've done this is by applying a superhydrophobic coating to these valves. So this is inspired by the lotus leaf in nature, which is a natural superhydrophobic surface. And this is work that we're doing with Arun Kota. And in this valve, we're applying a superhydrophobic coating to a commercially available mechanical heart valve. And this coating involves a microscale structure and a nanoscale structure that allows it to become superhydrophobic. I'll skip over some of the physics involved to keep the focus of the topic. But when we apply blood to the surface, the superhydrophobic surface is quite resistant to blood adhesion to the surface, whereas normal pyrolytic carbon, which is what these valves are made of, sticks to the blood quite readily. So you can see blood bouncing off on the right side of the superhydrophobic coating, whereas it sticks to pyrolytic carbon on the left. And similarly, if you flow blood down pyrolytic carbon or glass, it leaves a trail behind, which you can imagine then the blood's really sticking to the surface and is at risk for forming these blood clots. Whereas with a superhydrophobic surface, the blood just rolls or slides along the surface without actually sticking down to the surface. So this is all on the macroscopic scale, but then you might ask, well, you could begin forming a blood clot on the surface on the microscopic scale. So if we use a scanning electron microscopy, we can look on that scale and we can see if a surface is not superhydrophobic, so it still has microstructure but doesn't have nanostructure, you get a number of cells adhering to the surface. Specifically, there is inflammatory cells or leukocytes adhering, and then also platelets, which are the cells that really compose this blood clot or thrombus. Whereas when we have a superhydrophobic surface that has this also nanoscale structure, there's really no cells adhering to the surface, so the surface actually seems to be quite a good option in terms of resisting thrombosis on it. But in addition to surface interactions, you can result in blood flow inducing thrombosis or hemolysis. So as I mentioned earlier, high shear stress can cause hemolysis, so the rupture of cells, specifically red blood cells, or it can activate platelets, which then create this blood clot or thrombus. So when we look at this coating, we find that compared to FDA-approved valves, we don't actually increase instantaneous shear stress that would otherwise rupture these red blood cells. And we also don't alter the Reynolds shear stress, which has been fairly predictive of predicting blood cell damage. So that looks great. But when we look then more closely at the valve, we also can get leakage through these mechanical heart valves. So with these valves, you have two leaflets that flap open and close as the heart pumps. But when the valve is closed, there's going to be a small gap between the two leaflets and also between the housing of the valve and the leaflets. And with this small gap, what people have found is that's actually quite prone to thrombosis when the valve is in a closed state. And in this region, it's been found that actually shear stress can become an order of magnitude higher than what happens downstream of the valve. So this is also quite an important region of the valve to understand. So to look more closely at this portion, because it's difficult to look into the valve as blood's going through it, we also instead looked at developing a microfluidic device that allows us to look more closely at this blood reaction with these geometries. These devices have many advantages, including the fact that you don't require a lot of blood, so that's a nice advantage because you don't have to take liters of blood out of a volunteer in order to run the experiment. You can also match the shear conditions of the mechanical heart valve, so you can mimic really the mechanical environment that these cells are experiencing. You can also have strong control over the chemical environment that this blood's reacting at. So you can begin to look at specific ways that the blood's causing this thrombus to occur. There's also limitations because this material isn't the same as a mechanical heart valve, and also the Reynolds number is much lower because the dimensions are much smaller. So when we flow blood through this geometry, you can see platelets accumulating behind these geometries that are meant to really mimic these leakage areas around mechanical heart valves. So this video is really for PDMS, which is just a polymer that's commonly used for making microfluidic devices. And then eventually this platelet accumulation can result in a thrombotic response due to activation and formation of fibrin, which I won't get too far into the biology. But then when we apply the superhydrophobic coating by applying microscopic and nanoscopic polystyrene beads, we find actually that there's a reduction in the amount of thrombus growth. There's still a little bit of thrombus growth, but we're still optimizing this coating for these channels. And even at a larger scale, we've seen a very similar response from work I've done previously. So the gap size through this glass capillary is about the same as the gap size of a mechanical heart valve, and we can actually monitor thrombus growth. And we find actually the growth curve looks very similar to the previous growth curve for the microfluidic device. So the scale doesn't seem to be highly impacting the way that the thrombus is growing. And similarly, if you then normalize that growth by the surface area it's growing upon, you find that the growth rates are actually very similar between the two surfaces. So the thrombus process is happening very similarly in both cases. And unfortunately, if you look at the thrombus growth rate curve, you find actually that mechanical heart valves typically operate where you tend to get a lot of thrombus growth, whereas if you go down to lower shear stress, you get much less growth. So shear stress really seems to be controlling this thrombus growth. Again, it's the mechanical environment that's influencing disease. And so again, as engineers, what are the forces and timescales of this happening? Well, as the platelet's going through this constriction, it only has about 0.5 milliseconds or 500 microseconds to adhere down. And then once it's down, it experiences a drag force of about 2,000 piconewtons, whereas platelets can only have a single bond of about 100 piconewtons. So somehow these platelets are finding the surface and sticking down onto the surface. So it's much like a jet coming into an aircraft carrier and somehow landing on the surface. But in engineering, we have a lot of control systems that allow this to happen. And we also have a series of cores that then stop the jet. Platelets don't have that in theory, but actually they have something very similar in the body. So there's a protein that's in our blood known as von Willebrand factor. Again, I'll try to skip a lot of the biology behind that, but normally